Hi everyone, this is Anjum. Welcome to my channel. In today's video, we will discuss about one of the most familiar disease in the Karnataka region, that is Kyasanur Forest Disease. Let us know what it is. Kyasanur Forest Disease, which is shortly abbreviated as KFD, it is a tick-borne viral hemorrhagic fever endemic in Karnataka state, India. It is also referred as a monkey fever by the local people, that is called a Mangana Kaile in Kannada. The virus causing the disease is the KFD virus, which belongs to the genus Flavivirus and the family Flaviviridae. The KFD virus was first identified in 1957 when the illness occurred in the monkeys in Kyasanur Forest area around the Sagar and Sorba taluks in the Shimoga district. The case fatality rate of KFD virus infection is about 2 to 10%. Let us know the distribution of KFD. Initially, the disease was limited to 3 taluks, that is Sagar, Sorba and Shikaripur of Shimoga district of Karnataka. In subsequent years, the disease spread to the Uttar Kannada, Udupi, Mangalore, Chikmagalur, and in 2013, an outbreak of KFD occurred in Bandipur Tiger Reserve. This is due to deforestation and timber logging and encroachment of human activities are responsible for the increase in transmission and spread of KFD virus in new regions. And also, according to serological studies, evidence of KFD virus and related virus were found in different parts of India, such as west of Calcutta, West Bengals, Andaman Islands, and viruses related to KFD have been identified in China and Saudi Arabia. One question arises that why this KFD is only particular to the Western Ghats? Because the Western Ghats provides an ideal topographical climatological condition for the vector ticks, thus making the Ghats epitome for the tick-borne disease. Let us know the structure of KFD virus. KFD virus is ranked as a high-risk pathogen requiring biosafety level 4 handling, and it is a member of Flaviviridae and belongs to the tick-borne encephalitis virus. And Haemophilus spinagira have a diameter of 50 nanometer and which contains a 3 structural protein for the capsid and which contains a envelope and membrane. This virus enters the cell via receptor mediated endocytosis and undergo an immature and mature phase inside the cell. A recent study showed that infection of KFD virus is lethal in mice and leads to the elevated levels of pro-inflammatory cytokinesis in the brain. The virus contains a spherical positive single-stranded RNA molecule of 11,000 base pair in length. Okay. As we know that ticks are notorious as vectors of humans and other animals, they are most important vectors in the veterinary field and second only to the mosquito. Haemophilus spinagira act as both reservoir and vector for KFD virus, and it is a hard-bodied tick and widely distributed in tropical forest of peninsular India and Sri Lanka. It is an obligate ectoparasite of mammals of various rodents, insectivores and monkeys. It is a potential vector of Kyasanur Forest Disease. Haemophilus spinagira is a vector for the KFD virus, and these ticks have a life cycle lasting approximately 12 months and involving 3 feeding stages. In each stages of their life cycle, they suck blood from the vertebrate. The adult ticks occur during the rainy season and they have a relatively limited host range, and they attach on the larger animal and drop to the ground where they lie dormant till the onset of drier months. And adult females lays eggs and which molt into larvae. The fed adults are deposited in the rainy season in the forest floor and eggs laid in the cluster. The eggs hatch into thousands of larvae and they have a limited mobility and they have picked up by the small moving animals and large mammals all in the dry season. The fed larvae drop to the ground and molt into nymphs. These seek the host, mostly small animals, birds and man. The fed nymphs again drop to the ground and molts into adults and which attach to the new host are mostly large animals. In the life cycle, the cattle play a very important role in KFD epidemiology, though the virus does not circulate to any significant levels in them, but they are the main multipliers and distributors of vector population because cattle roam all over the forest. And KFD virus is transmitted by an infected tick, especially nymphal stage ticks. The wild monkeys Semnopithecus entellus and Macaca radiata gets the disease through the bites of infected ticks. The infection causes severe febrile illness in most of the monkeys. When the infected monkeys die, the ticks drop from the body and thereby generating hotspot of infectious ticks that further spread the disease. Humans get the disease from an infected tick bite or by contact with an infected animal such as sick or recently dead monkey. The available epidemiological data does not suggest any human to human transmission. Pathogenesis model of Kyasanur Forest Disease. The virus enters in body on tick bite or through contact with an infected animal. The virus initially targets mainly macrophages and dendritic cells, as you can see in the picture. The multiplication of virus in these host cells yields high viremia and leading to systematic spread of the virus to the spleen, liver and other replication sites to produce disease symptoms. Mainly they attack dendritic cells and macrophages. Later they spread to liver and other replication sites. The infected antigen producing cells, that is APCs, that present the viral antigen to T cells could release large amounts of pro-inflammatory cytokines early after infection and also modulate host immune response via type 1 interferon production. The antigen positive T cells could also produce interferons 1 and subsequent activation of JAK-STAT signaling induces an antiviral state for elevating virus burden. The humoral immune response via production of antibodies by activating IFN response by inhibiting JAK-STAT pathway, possible bringing about uncontrolled viral replication and poor immune system. The multisystematic illness might be attributed to the pro-inflammatory cytokine storm that could contribute to immunosuppression and disease progression by inducing neurological complications and vascular dysfunction that leads to hemorrhagic manifestations, multi-organ failure and shock. These complications altogether finally result in death of human. Let us know sign and symptoms. Incubation period estimated to be between 3 to 8 days after the bite of an infected tick. The symptoms are sudden onset of high-grade fever with chills, intense frontal headache and body pain, photophobia, vomiting, diarrhea, and respiratory symptoms like persistent cough in few cases. After 3 to 4 days, the symptoms such as a severe muscle pain, low blood pressure, low platelet, low WBC, RBC count. After 1 to 2 weeks, the second stage shows neurological problems and mental disturbances, etc. In few cases, hemorrhagic symptoms may occur in early stage in the form of bleeding from the nose, gums, etc. Some patients have persistent cough with blood-tinged sputum. Untreated cases rapidly progress to the coma, even to death. A post-mortem findings revealed predominance and flooding of macrophages and lymphocytes in the liver, spleen and kidney. A diagnosis. The designed laboratory for diagnosis and isolation of KFD virus is National Institute of Virology, which is situated in Pune, and human blood samples, monkeys, viscera, ticks are collected from the field and they are tested for KFD virus. The laboratory techniques which involves a detection of KFD virus by real-time RT-PCR technique, and this is the first line for the test of diagnosis of KFD, and it can detect the virus in the sample. The second technique is the ELISA test. Prevention and control. There is no specific antiviral drug therapy for the KFD. A supportive treatment with the maintenance of proper hydration in the body, a formalin inactivated chick embryo fibroblast vaccine developed in 1990s is currently licensed and available in India. It is given in 2-dose schedule followed by the routine boost, and spraying of insecticide may be carried out in the areas where the monkey's death have been reported, and vector control may be done by dusting with insecticides or by spraying with pyrethroids. And very important step is personal protection and application of tick repellents such as dimethyl phthalate, encamine dimethyl M-toluidine, and people living in the forest area should strictly use tick repellents and protective clothings, and health education and illegal fencing of forest trees and deforestation need to be ensured, and regular activities to be conducted by the field staff to educate the people about the disease and make them to accept the vaccine. Let us know sign and symptoms. Incubation period estimated to be between 3 to 8 days after the bite of an infected tick. The symptoms are sudden onset. And briefly we will sum up the topic. Kyasanur Forest Disease, which is shortly abbreviated as KFD, it is a viral infectious bleeding disease in the monkey but also affects the humans. The KFD virus was first isolated from sick monkeys captured in the Kyasanur Forest in the Shimoga in 1957. Hence it is also known as Kyasanur Forest Disease or monkey fever. KFD virus zoonotic in origin and transmitted by infective tick, that is Haemophysalis spinifera. During the period of 1957 to 2004, a total of 24,721 human cases and 6,475 monkeys deaths were recorded from Malnad belt of Western Ghats. KFD virus is controlled by vaccination, tick repellents, wearing protective cloth, health education and personal protection. These are my reference. Thank you. If you like the videos, please like, share, comment and subscribe to my channel.